So Isabella Oyier is the head of Biosciences at the KEMRI Wellcome Research Program in Kilifi, in Kenya. And she's a longtime friend of the malaria gen community. She's been involved with genomic epidemiology for a very long time. Of course, that makes her sound old. She's not, but you know, that's. But so she's been collaborating with us for a long while, and she's a very very strong genomic epidemiologist. She actually ran she ran the COVID surveillance project during the pandemic, and she's doing some genomic surveillance work in Kenya for malaria. So she's kindly done a recording of a talk about her work.
Good afternoon, and thank you to the organizers, especially Olivo and Varanya, for this opportunity to present, even though virtually. It's a real pleasure to be one of the presenters at the GenRe-Mekong Annual Scientific Forum. I'm going to take you through the work I've been doing on genetic surveillance of malaria in Kenya. So this work originally started with a collaborative project between my institution, the KEMRI Wellcome Trust Research Program, my group, and colleagues at the KEMRI program in Western Kenya in Kisumu, where we used their Sentinel Health Facility sites to make an assessment of the KELT13 mutation frequencies in samples collected between 1995 and 2003. This is before the introduction of artemisinin combination therapies. And we showed that most of the parasites across the country were wild type, represented by the dark gray bits of the pie chart, whereas the light gray bits of the pie chart represent the mutations we found. And just to be clear, that we didn't find any mutations that conferred resistance to artemisinin. So my group went on to do a longitudinal study to take advantage of the fact that since the mid-90s to present day, we've had 3 changes in our national treatment guidelines for malaria, and we assessed the chloroquine resistance transporter gene, the marker for chloroquine resistance, and showed by the red line, which is a chloroquine resistant genotype, and the black line, the chloroquine sensitive genotype, that at the peak of chloroquine treatment failure, that was the height of the chloroquine resistance genotype. And when we shifted treatment to sulfadoxine-pyrimethamine, and then later to the ACTs, Coartem as our first line, there's been a dramatic and drastic drop in the drug resistance genotype and a re-emergence of the sensitive genotype. And we use both these data sets to engage with the National Malaria Control Program on how important malaria molecular surveillance is and what it allows you to do with regards to tracking drug resistance mutations over time and in space. And then that allowed us to start a conversation and through a co-creation process, come up with the idea of developing a national malaria molecular surveillance. And this was for beyond research, but more for public health action. So we were thinking about terms of reference, the review of data, that could inform some questions we would like to ask, where we could use malaria molecular surveillance. And this we would do collaboratively between the 2 institutions. And so the 2 areas that were identified by the National Malaria Control Program were diagnostic resistance and drug resistance. And through the project, the aim was to set up a technical working group for malaria molecular surveillance within the NMCP framework, develop some sustainable malaria molecular surveillance tools in my lab, and some user-friendly bioinformatic tools so we could provide some actionable data for decision-making, public health decision-making. And then, together with the NMCP, conduct malaria molecular surveillance at a national scale. So diagnostic resistance is important in our region, and this is because particularly in Eritrea, where they've made assessments of the protein, which is detected on the RDTs, the histidine-rich protein 2 and the histidine-rich protein 3, which is a cystogene, that they're at a high frequency of these mutations in Eritrea, greater than the WHO threshold of 5%. Similar studies have been done in Ethiopia, not yet hitting the threshold, but I think fast approaching from our more recent data, And Uganda have done a similar study, but their frequencies are quite low. So Kenya is working with the WHO. They've just finished the sample collection for the assessment of HRP23 deletions in the country, and we're collaborators on that project. For artemisinin partial resistance, this has now emerged and has been has emerged independently from what's been observed in Southeast Asia, where in Rwanda this was first described in a 2020 publication with the mutation R561H, and later the C469 and A675V in Uganda, and later the R622I in Ethiopia and Eritrea, and more recently published this year in Tanzania, the R561H. And all these studies have demonstrated that these mutations are associated with delayed parasite clearance. So that means in the clinic, they were seeing individuals who had parasites with these mutations still had parasites on day 3 after treatment. So both these areas both these drug resistance and artemisinin partial resistance are a cause for concern in our region. So Kenya is using the National Malaria Molecular Surveillance Platform as an early warning system. And that's what we're aiming to roll out with this project that is funded by the Bill and Melinda Gates Foundation. So we have developed an amplicon sequencing panel on the next generation sequencing platform called an Illumina MySeq. And what this allows us to do is in regions such as ours with high malaria transmission, an individual will be infected with several genetically different parasites, and we're able to detect these genetically different parasites represented by the colors here using this amplicon sequencing pipeline. And so in our panel, we've included KELT13, the artemisinin resistance mutation, dhfr, the pyrimethamine resistance mutation, dhps, the sulfadoxine resistance mutation, and MDR1, which has 2 mutations that have been rising in frequencies since the introduction of artemisinin combination therapies. We do have another marker called, which has very many mutations in it, called apical membrane antigen 1, AMA1, that allows us to really clearly define these different parasite types within an infection in our setting. And so we tested that panel on samples we obtained from the school surveys in Western Kenya. These are 8 counties around Lake Victoria. And within each of those counties, we collected from 10 schools, 100 samples each. So in total had about 8,000 samples with 28% positivity by RDT. And these samples were collected in 2019. And what we show for pyrimethamine resistance, that there's a high level of resistance with about 3 mutations at 78%, a high level of resistance to sulfadoxine, again with 3 mutations, at about 66%. But for artemisinin, a low level of resistance, 7.7, but even so, it's not resistance, it's just we've identified a mutation, and these mutations do not confer resistance. So there's a low level of frequency of mutations. And 91% of the parasites, as at 2019, were wild type. So in 2019, from asymptomatic malaria samples, there's no cause for concern for artemisinin resistance. We then used the same panel to do a health facility surveillance study, where early this year, there was a suggestion that within one of the counties in Kenya, Kisii County, based again in the West, that there might be the R561 mutation that was described in Rwanda. So we collaborated with the NMCP, who had some samples they had collected from this county in 2022 as part of the diagnostic resistance study, collaborating on them with. And we did Sanger sequencing in the first instance, because this is what had been done in the earlier publication, and then went on to use our next generation sequencing amplicon sequencing tool. And with the old method, there are no mutations of concern. In the new method, we do identify loci that may confer resistance, but in this particular sample set, this is the location that was identified in Rwanda, Uganda, and this position in the K13 gene has been identified to confer resistance in Southeast Asia, confer resistance to artemisinin. And so at this point, there's no cause for concern because there's no change in the protein, KELT13 protein, but it does highlight that surveillance should be continued and should become routine. And so we shared this as a report in this policy brief with the NMCP. For diagnostic resistance, we have studies ongoing in Kilifi in primary healthcare facilities, where we do RDT measurements as well as microscopy measurements, and we identified those that were discordant, so RDT negative and microscopy positive, about 11 of them. And none of these samples had none of the samples were HRP2 negative and HRP3 negative. We increased the sample size using real-time PCR, a more sensitive method, and where we can quantify those parasites per sample to about 5 parasites per microliter. And then we did the assessment and found that the HRP2 negative, HRP3 negative was actually at about 2% or less than 2% in the population. So still very low, but we have to use, so this method was done using maybe a less sensitive technique, and now we're improving our technique using a more sensitive technique developed at the London School of Hygiene and Tropical Medicine, Khalid Bashir's lab, where we're able to show, as an example, that in this sample, that's 2D7, but also as a human sample, that you can detect all the markers. So the blue line is the HRP3 gene, the red line is the HRP2 gene, and just showing there's some human DNA in there. And the green line is another parasite marker. But when you look at the parasite, this is a parasite from the laboratory that does not have the HRP2 gene. We're not able to detect it using the assay. And this is a parasite that does not have the HRP3 gene. So we don't detect the blue line, we detect the red line, that were quite low using this assay. So we still have a lot of work towards optimizing this assay further, and then we will be able to roll it out to support malaria molecular surveillance and the national platform. So we are now working towards the National Malaria Molecular Surveillance Platform. We have developed a database of data that has been published in Kenya since 2000 for drug resistance markers, and we're using this database as well to now input any more recent data that we're producing into this database so that it's available to the National Malaria Control Program. We're working on a technical working group to be within the NMCP structure and a strategy document as well. And this would be both in the NMCP structure as well as within the National Public Health Lab, where they have a pathogen genomic surveillance unit. And we're hoping that this document sits within the national framework as a strategy. We have run trainings for our National Malaria Control Program and the National Malaria Reference Lab colleagues who've come about 4 times to our laboratory to learn PCR and sequencing and DNA extraction, and so that we can help them understand what the outputs are likely to be once we set up the National Malaria Molecular Surveillance Platform. And what we've done so far with regards to developing this platform is looking to about 14 counties across the various malaria endemicities demonstrated by the different colors. So the lake endemic is the orange over here, and the coast endemic is yellow. And we're looking to establish sentinel health facility sites in these 14 counties. We have begun our engagement with these counties, indicated by the green ticks. So these are about 6 counties we've engaged, and they're quite keen to work with us to start the diagnostic and drug resistance surveillance plan. We have shared with them as well the criteria for how one would select a sentinel health facility, as well as the data information sheet or case investigation form that would go alongside the dry blood spot collection. So this is the current plan that we've engaged the counties with, that they become a sentinel health facility site, they identify malaria positive patients, and when they're taking the blood sample to confirm the RDP positive, we'll get a dry blood spot as well, and the malaria case investigation form filled out. Both of these will then be shipped to our lab, where we'll work in collaboration with the National Malaria Reference Lab to do the sequencing, and then we'll produce some policy briefs and reports that will be shared with the NMCP as well as the sentinel health facility sites. the overall aim of producing a national map for drug resistance and diagnostic resistance, as well as a longitudinal map so that we can continue to track the mutations that we identify over time. And so this is the plan for here would be the dry blood spot sample collection, and then these get shipped to our lab and how we'd receive them and enter the data, do the extraction of the DNA, then move on to PCR and sequencing and the bioinformatic pipelines to then produce the policy briefs as well as some collaborative manuscripts. We have projected maybe a 14-day turnaround time from receiving samples to sharing results, but this has to be tried and tested once we actually start the sample collections in these counties. And the projections at the moment is for a start next year, particularly with the 6 counties we've already engaged with this past month. And so I'd like to just thank my team that have been working on the molecular tools, the bioinformatic pipelines, so that we can work towards developing the National Malaria Molecular Surveillance Platform, the National Malaria Control Program, the Ministry of Health, the National Public Health Lab, our colleagues in KEMRI, who have been doing the school surveys, so they go out and do a deworming project, and we piggyback and give them some DBSs and RDT so we can collect these dry blood spot samples from asymptomatic individuals. Our collaborators in the US, who've been helping us optimize the laboratory assays as well as the bioinformatic pipelines, and then PMI and CDC, who we work very closely with on some therapeutic efficacy studies and where we're able to also demonstrate the power of the amplicon sequencing pipeline using these sample sets. And finally, the funders from the Bill and Melinda Gates Foundation that have allowed this project to become a reality in Kenya. And thank you all for listening.
Actually, I thought it was quite interesting how there are many similarities and many differences with respect to things that are going on here, but you could see a lot of things very much parallel to what we've done in this region. So, unfortunately, Isabella is not connected to answer questions, but if you've got questions, please give them to us. Okay.